You perhaps don't know that half of the oxygen we breathe derives from plants, bushes on land, and the other half derives from algae, the microalgae, because in the water of the ocean, of the lakes, there are unicellular organisms that have the same function as plants. They photosynthesize, so they produce oxygen, they fix carbon, the oxygen we breathe. So they are invisible, we know very little, we'll talk about more, but they play a very important role, and they are at the base of the food chain in the marine environment. So they are the food, as on land the grass is eaten by the cows, by the insect, the same at sea. Those tiny microalgae are eaten by the little crustaceans and so on, up to the fishes. A very important role. The same also for the big algae that grow in shallow water, because those are plants, they need light. So either floating in the water or attached to the rock. But the invisible one floating in the water are very abundant and poorly known.
So poorly known, and such a large community of scientists is doing all that. How comes that it is so unknown?
So first of all, the marine ecosystem is much more difficult to access than the terrestrial one. We have to have ships, and we usually use research vessels to go out to the ocean, from the coast to the open ocean, and then we need to sample all the microalgae, and they live usually in the surface of the ocean, but they are distributed over all the oceans, including polar oceans such as the Arctic and the Southern Ocean. And these ecosystems are particularly difficult to access because they have seasonal sea ice cover, so you have to have icebreakers to go there and to sample and collect the algae. And once we have collected them, we bring them to the lab and then we study them. As Marina said, we look at their diversity, and they're very, very diverse. They're microbes, they have large populations, and the marine system is very dynamic, so they can drift with currents. And they multiply, they form blooms, specifically in spring, when there is enough light and there is still enough nutrients after the mixing of water in the winter. And then they form large blooms, and these blooms can be as large as the UK, for instance. And we have two major groups of algae in the ocean, microalgae, diatoms, which live in glass houses. And they contribute significant to the global carbon cycle, but also they underpin important food webs. They're important for fisheries, for instance, because fish feed on them. And then also the coccolithophores. You have certainly heard of the White Cliffs of Dover in the UK. And the White Cliffs are basically made of microalgae because they calcify, and this calcium carbonate has formed these massive cliffs of Dover.
So they can give both the white color and the greenish color of the sea.
Yeah, because they have pigments to harvest light and to photosynthesize.
In the attempt to approach them, to be able to study them, you now these days you have a lot of technology, and can you tell us please about the institutes that you work in? How do you do that? What kind of equipment do you have, and what is, for example, the usual study trip or research that you do?
Okay. Yes, I work in Napoli, in Italy, along the Mediterranean Sea. So our is an old institute that was founded in 1872, and most of the people work on marine sciences and the group on marine microalgae. In Napoli, we have a long-term station. What is that? A place in the Gulf of Naples where we, every week we collect samples. This allows us, since 1984, to have a sort of a movie of what's going on, to reconstruct the seasonal cycle of the algae. Who is there, how big the blooms are, who is eating the algae, what are they doing when we do not see them. So besides following what's going on a temporal scale, we also study selected species. Thomas mentioned the diatoms, which are very important, not only in the you know, in the polar regions, but also in coastal waters. So we establish cultures, that is, we pick the individual cells that they grow in the lab. So we have labs, we study their diversity. How is done that? We examine these structures in electron microscopy. You might see in this day here in Zagreb very nice exhibitions with the pictures in scanning electron microscopy of coccoliths. So they are really beautiful, but besides the beauty, we want to see which are the characters that distinguish the different species. So we study their morphology, but now we also have molecular tools, sort of molecular barcodes that are characteristic for each species. And so that's, in a way, we also study the life cycle, because it seems strange, but these little tiny cells, these little tiny balls, have also very complex life cycles. They produce resting stages, they sink to the bottom, they can have a sexual life as well. So to understand, we know a lot about trees, no? When they have fruits, when they flower, if they have parasites, viruses, no? Those topics are very common, belong to our daily life. However, for microalgae we know very, very little about, but we start now exploring, and maybe Thomas, in their institute, they do very, very interesting and advanced studies on the genomic secrets of the microalgae. That's another topic that has become very important to understand their biology.
Yeah, so I'm from the University of East Anglia, which is in Norwich, and Norwich is about 2 hours northeast of London. And this is a relatively young university, but one of the major strengths is that we are very interdisciplinary, especially in the school where I am based at, the School of Environmental Sciences. We do research from the molecular area to social sciences, and we try to integrate the different streams of knowledge in order to come to new knowledge and to advise government. This is also very important. This is what we do in the UK. And what I do specifically, as Marina said, is to look more at the genomes of microalgae. Like any other organisms, algae have DNA, and the DNA encode for proteins, and the proteins make the phenotypes. They're responsible for their metabolism, they're responsible for how they grow and how they respond to ecosystem changes, which is, in the context of global warming and global change, very, very important. And so what we do is, in order to get a better idea about how they have evolved and how they respond to a global change, we sequence their genomes and we look at how the genes are being regulated under important environmental conditions. One is, for instance, ocean acidification, because we put too much CO2 in the atmosphere by burning fossil fuels. All the CO2, or most of this CO2, is ending up in the oceans, because oceans cover 71% of the surface of our planet, and this impacts the pH. And if the pH goes down, then some microalgae, like the cucullitophores, they struggle because they calcify, and calcification takes place at higher pH.
So there is no food?
This could be one consequence. Another important issue is that due to global warming, the ocean is more stratified in certain parts. And this stratification has an impact on the nutrients available for microalgae. So if the water column is not mixed enough, then the nutrient concentration in the surface layer, they are being reduced, and this is not good for phytoplankton. If phytoplankton can't grow, then there's no food available for the marine food webs.
So you have a lot of equipment. What kind
Yes.
of?
Yeah. So we have, in the UK, we have access to several ships. We collaborate with the National Oceanography Centre, NOC, in Southampton, for instance. We have close by CFAS, and they all have ships. So Norwich is not as close to the ocean as Naples, but we are about 40 minutes away by car. And whenever we need to, we use ships in order to go out, in order to study the ecosystem. I have done this extensively for the Arctic and specifically the Southern Ocean as well, because the Arctic is suffering from global warming the most currently. We are very, very concerned about this ecosystem, and therefore there are global initiatives to try to understand the ecosystem better, because as I said in the beginning, it's really difficult to access this ecosystem. We have darkness for several months in winter, and then we have 24-hour light in summer, and we have seasonal ice cover, so you need icebreakers, so this is all very, very expensive. You need to have good ships in order to access these ecosystems. But there are global initiatives. Because this is so important, and because we don't know much about the organisms specifically in the polar regions. And,
so how often do you go there?
So, you know, when I was younger, when I was a PhD student and a postdoc, I had more time, and I went 3 times to the Arctic Ocean and 2 times to the Southern Ocean with icebreakers from Germany. I studied in Germany, and we went to the Arctic. I spent several months in total in the Arctic, and we looked at microalgae that live in sea ice. This is a very extreme environment. You know, it's frozen seawater, but it's not as solid as freshwater ice. It has channels inside, and inside these channels, microalgae can thrive. And it's a very fascinating environment. They are highly adapted. They can grow at -10 degrees Celsius, and they do not require a lot of light. Only very, very low light is required to grow and to form massive blooms. This can happen in polar regions too. And this is why, for instance, the Southern Ocean is so rich in life. There are a lot of whale populations and seal populations, penguins, of course, everybody knows penguins. And this is this boils all down to phytoplankton, to microalgae that flourish in the surface of the ocean.
So we can say that your research is also very, very beautiful.
Yes, that's right. Yes.
Can you share with us some of the major results that you achieved by now? Maybe, yes. Just a second. Yeah, like this.
Yes, I think, yeah, maybe as for the group of people with whom I work, an interesting result is what I was mentioning before, to study the, let's say, the life cycle of this organism. We selected an interesting one, because what we maybe didn't mention up to now is that some of these microalgae produce toxins. As plants, we have toxic plants, and we have toxic microalgae, which is obviously relevant for the fact that we then eat mussels, and those mussels might accumulate toxins. So, to make the story short, yeah, our selected species that we studied that is one of these diatom, toxic diatoms, and we studied the life cycle of these organisms, both from the, say, morphological point of view, but also the molecular point of view. We found out that there are males and females, plus and minus. And obviously, as with many animals, for example, if for a certain reason you don't have the plus or the minus in the environment, this algae disappears. So which are the mechanisms that are at the base of sex regulation? And this is an interesting piece of work that was produced in recent years. So to understand the mechanism of this microalgae, but also to follow them in the natural environment, thanks to the long-term station I was mentioning before, it's possible for us to collect samples over time and to study the level of diversity that's within a certain species. Are all the cells the same? How different they are as related to their molecular profile, but also of their performance, how much toxic is produced and things like that. To go deeper in the biology, few are the species that are studied at that level, I would say, but people start. In Tomas' lab, they studied a lot one species that lives in the ice, and there are a few species for which these approaches are possible. But they are important because you have to understand the basic biology of a species in order to understand why it's there, why it produces a bloom, and things like that.
And also maybe to compare.
Absolutely. Yeah. So my group is very interested. My group is very interested in trying to understand how the environment impacts the evolution and adaptation of microalgae in the ocean. And what we do, we try to bridge gaps from large scales to subcellular processes. And I can give you one example. So what we did over the last couple of years is we went with one of the big icebreakers and ships available from the Arctic Ocean down to the Southern Ocean. And we sampled about 80 locations along the transect from the Arctic down to the Southern Ocean in order to understand what are the main environmental drivers impacting and determining the biodiversity and activity of phytoplankton communities in the global ocean. And what we found by this comparative approach, because you do not only, when you do a study like this, it requires to look at a lot of different parameters to make sense of your specific microalgae data. So we measured nutrients that are in the water, we measured light, CO2, temperature, and by comparing different environmental variables with the presence and absence of genes, with by looking at how these genes are regulated under different environmental conditions, we found that temperature is the most important environmental variable impacting the diversity and even metabolism of algae.